Video, I will be talking about biomedical electronics and biosensors. But first, let me introduce what is an electronic engineer. Um, Electronic engineers design, develop, and oversee the production of electronic systems and products. And with these products and their components is a part of the process of making sure of all electronics work efficiently, but not just efficiently, also reliably for personal and commercial use. So, let's talk about biomedical electronics. Did you know that biomedical electronics is a specialized technology that focuses on the intersection of electronics and biology? With the developing technologies and devices, it improves the healthcare, diagnosis, treatment, and also monitoring of medical conditions and the overall health of a person. With this, it undergoes the principles of electronics, biology, physics, and medicine, which creates innovative solutions with with the medical challenges and deepened understanding of the biological systems under electronics. So, in this part, it is the diagnostic equipment. Um, Here are the samples. We have ECG machines. ECG machines records the electrical activity of the heart over time, which our heart produces tiny electrical signals when it beats. And these beats are signals that travel through our body. We also have the electrodes and sensors, which are placed either in our chest, arms, and legs. Usually, the conductive pads, often called or the silver chloride, it picks up the electrical signals from the body. We also have the amplification and filtering the heart's electrical signals. Um, it is a very small electronic amplifiers boost the signals while the filters remove the noise. We also have the digital conversion or analog to digital conversion. After the amplification and filtering, the signal is digitized, which allows the ECG to display on a screen or save as data. We also have the microcontroller or processor. It processes the data to detect heart rate, etc., and also can run software for signal analysis and display. You can see the chemistry side of the electrode chemistry or the silver chloride, which is pretty common or most common. The silver reacts with chloride in a gel to form a stable half cell. It ensures the consistent signal capture and minimizes the drift. We also have the electrolyte gel. It is used to reduce skin electrode resistance. It also contains ions which make the skin more conductive. We also have the skin interface chemistry, which is the outer layer of skin, which is the stratum corneum that is resistive, which helps the gel to break down its resistance. Next, we have the MRI scanners. MRI is magnetic resonance imaging, and this type of technology, it produces three-dimensional detailed anatomical images. It is used for disease detection, diagnosis, and lastly, the treatment monitoring. It is a perfect storm for electromagnetism, quantum physics, electronics, and chemistry. We have for the electronic side, the MRI is of course, electronic machine. It is a powerful magnet which produces a strong magnetic field that forces protons which the body align with that field. When a radio frequency current is then passed through the patient, the protons are stimulated or spin out a equilibrium, straining against the pole of the magnetic field, and if the radio frequency field is off, The MRI sensors are able to detect the energy released as the protons realign with the magnetic field. For this, the chemistry side is the hydrogen atom chemistry MRI, which is the hydrogen nuclei or the lipids. The chemical environment affects how rapidly excited protons go back into equilibrium. The T1 and T2 relaxation times are the key to contrast. It also influence actually the molecular structure, binding states, and the tissue environment. With this, the contrast agents, chemically engineered molecules which contains the gadolinium shortens T1 relaxation time, which makes the tissues appear, like you can see, like brighter, and also must be the brighter it is to avoid the toxicity. The new agents that are being developed, it targets the molecular imaging, for example, tumor specific agents. With this, the tissue biochemistry inside MRI, it can detect chemical shifts, fat versus water content. Next, we have the basics of biosensors, but let's talk about the bioreceptor. Bioreceptor is a biologically derived molecule such as enzyme, antibody, nucleic acid, or cell that specifically recognizes and binds to a target substance, or called as the analyte, which triggering a measurable signal used in biosensing applications. This recognition is super precise since it gives biosensors the ability to detect things like toxins, pathogens, or important biomolecules. Next, we have the transducer. This type of element converts a biological signal originating from a physical or chemical change caused by the interaction between a bioreceptor and an analyte into a measurable output. This measure of signal can be categorized based on its nature, such as optical, electrochemical, or piezoelectric. These signals are then processed and displayed, providing information about the analyte. Now we have the signal processor. In signal processor, it is an important element that converts signals from the transducer into useful information about the substance being detected. Did you know that the process information is then presented in a clear or easy to read format, helping people make quick, accurate decisions in areas such as healthcare. Next is the chemistry behind biosensor. It is important for ensuring reliable signal generation from the physical or chemical transducer. This can be categorized into biorecognition mechanisms. Next is the surface chemistry and signal transduction chemistry. Biorecognition mechanisms, these are the chemical interactions that occur between the biosensors, bioreceptor, and the analyte. These mechanisms determine the accuracy of the biosensor. For the enzyme substrate interaction is highly specific binding which occurs between an enzyme and its corresponding substrates within the enzyme's active site. This interaction is actually fundamental to enzyme catalysis, which is driving biological reactions with remarkable efficiency and selectivity. The example for this is glucose, or used in glucose biosensor. Next we have is the antibody antigen binding. This one is frequently or mostly used specifically in particular antigen like various bacteria or protein. For example, COVID-19 test. We also have the DNA hybridization, and this is a mechanism that relies on the complementary base pairing between nucleic acid strands, or actually a single-stranded DNA probe on the sensor binds to its complementary DNA or RNA target. Next, we have the hydrogen bonding, baroreceptor analyte binding. This one is when an hydrogen atom attached to an electronegative atom like oxygen or nitrogen. It interacts with another electronegative atom on the opposite molecule. Actually, this type of bonds helps to make the interaction specific as they form in certain parts of the molecules, allowing for selective recognition of the biosensor. The electrostatic interaction in baroreceptor actually involve attraction or repulsion between charged parts of the molecules. This called is or the dipole between polar molecules. Next, we have the hydrophobic effects in baroreceptor. In this part, it is non-polar parts of the baroreceptor and analyte which sticks together in water or in H2O. This happens because it reduces the disturbance to water molecules, making the binding, like more stronger, or making the process more favorable. Next, we have the surface chemistry. This is essential for attaching the biorecognition element to secure the transducer. The immobilization on the bioreceptor, the sensor surface of these unique techniques like physical absorption, covalent bonding, cross-linking, or entrapment. This actually ensures that the enzymes or the antibodies DNA probes remain stable or active during the detection. The surface functionalization discerns sensor surface by attaching specific functional groups such as carboxyl, thiol, or epoxy groups to enhance its interactions with baroreceptor. The signal transduction chemistry, in this, the biochemical between baroreceptor and analyte is converted into measurable signal. In electrochemical detection, it measures changes in current, voltage, or impedance that occur as a result of a biochemical reaction. Optical detection changes in light properties or surface resonance when the analyte binds to the bioreceptor, which enables real-time detections of interactions. The mass-based detection, in this one, it approach measures changes in mass, the biosensor fabrication. In this, the common materials used is actually, it includes metals such as platinum and gold or semiconductors. The nanomaterials have a unique set of properties that make them well suited for biosensors application. For example, gold nanoparticles. These are the tiny gold specks that changes colors when the target binds. Next, we have carbon nanotubes. This one is it conducts electricity very well, using sensors for detecting gases and biomolecules. Third one is the quantum dots. Quantum dots are semiconductor nanocrystals that emit different colors of light, but it depends on their size. Next, we have the nanowires. Nanowires are extremely thin wires made of materials like silicon or zinc oxide used for highly sensitive detection of proteins and viruses. Next, we have the conductive polymers. This one, it enhances the biosensors to perform through their ability to conduct electricity or more efficiently. The more sensitive the signal detection is, it leads to sensing the elements. These polymers facilitate the easy attachment of biomolecules, increasing the sensor's ability. Lastly is the hydrogels and smart materials. In smart materials and hydrogels, it includes hydrogels change their properties like size, color, or conductivity when it is being exposed to things like temperature or specific molecules. How can actually biosensor materials affect sensitivity? Let's say that the materials are used in the biosensor directly impact its sensitivity, specificity, and stability. Actually, the signal transduction also relies on the material choice. For example, how a biological event becomes a measurable signal. In highly conductive materials boost electrons flow in electrochemical biosensors interface materials this topic is it talks about how can a biomedical electronics actually helps, how biosensor actually works.